Okay. Okay, so let's we're gonna begin again with the amino acids. So the amino acids are part of our micromolecules. So we have micromolecules and macromolecules. We have micromolecules and so we have micromolecules. So we have micromolecules, and then we have macromolecules. So we build up from micromolecules into macromolecules. And this is not just happening with one type of micromolecules. We have amino acids. These are amino acids. These are the micromolecules that give us macromolecules known as proteins. So the amino acids are micromolecules that we build them up to give us proteins. Other types of micromolecules are your nucleotides. Your nucleotides are micromolecules that we build them up to give us nucleic acids. And we have also the lipids, and these are known as glycerol and free fatty acids, and we build them up to give us lipids. So these at the top are my macromolecules. These at the bottom are my micromolecules. There's a fourth one, and that is what we're going to begin the next module, the next exam 3 that was supposed to be exam 2, but this is what's going to be about. These are your carbohydrates, and these are known as monosaccharides that we build up to give us polysaccharides. So the names in the books are different. Sometimes they call them micromolecules. Sometimes they call them monomers. Sometimes they call them macromolecules. Sometimes they call them polymers. Sometimes they call them polymers. Monomers, polymers, micromolecules, macromolecules. If I am going up, if I am building up, I'm going from small to big. That is an inverted pyramid. These guys are very big, and these guys are very small. So we stack these micromolecules to give us the macromolecules. If I am building up, that is known as anabolism. So for example, I'll ask you the first question on Wednesday. When we are building up amino acid micromolecules into protein macromolecules, polymers, from monomers into polymers, what is the name of the process? Is it anabolism or catabolism? So the answer is anabolism. Or maybe a true or false. Building up amino acids into proteins, that's an example of catabolism. So the answer is false. Yes. The macromolecules for the lipids, the micromolecules. So if you are small, you are micro. If you are small, let me record this because if you are, these are free fatty acids. These are free fat. I'm gonna post this online. Yeah. Come back over to the small version. Come back over. Okay. Can I put it here? Is it dehydration? Yeah.
That's an apple.
Yeah. And you can go backwards, as well. Water.
Okay. All right, guys. So I'm gonna post this. So I forgot to start it. Okay. So we have monomers and we have polymers. We go from we have different types of micromolecules, and as a result, we will have different types of macromolecules. We were concerned with 2 out of the 4 for our first midterm, the amino acids and the nucleic and the nucleotides. So, but we must first remember before we go into the structure. So we're always studying the structure and the function. So before we go into the structure, we have to have a roadmap that this is building, we're either building up or breaking down. So why are we building up? That is anabolism. We are building up for storage. We are building up for storage. To store what? To store the energy. Because if I have a meal with a lot of proteins, my digestive system does not do anabolism. My digestive system will never store. My digestive system breaks down. But after it's broken down and absorbed into my blood, then my tissue will decide what is happening. Am I running the marathon? I need energy. So I'm going to be going in the opposite direction. Now I'm going to break down my macromolecules into micromolecules, and the energy is going to come out. So now ATP, I'm going to get the energy to come out of the breakdown. So I break down to get energy, and I break down the bonds. So when we look at the structure of the amino acid, and I look at the structure of the nucleotides, an example of a nucleotide would be ATP. So ATP is a nitrogenous base that is adenine. That's an example of a nitrogenous base, adenine, attached to a sugar, and attached to the sugar, I have PI1, that's the phosphate group, the first one. I have PI2, that's the phosphate group, the second one. And I have PI3, that's the phosphate group, the third one. If I want to store energy, I add phosphate, 1 phosphate, 2 phosphate, 3 phosphate. If I want to use energy, I go down this route, I break the bond. I break this bond, energy comes out. I break the second bond, energy comes out. I break the third bond, energy comes out. So the process of breaking down, that is when I point downward. When I point downward, that is known as catabolism. When I point downward, that is known as catabolism. When I break down, that is known as catabolism. Catabolism. When I break down, that is known as catabolism. I break the bonds of the nucleotide. So the energy comes out. So the energy comes out. That is a type of an exergonic reaction. Delta G negative, because the energy left the bond. So this delta G, this Gibbs free energy minus delta G represents, so this endergonic. Remember we mentioned that in the past. Endergonic. What is this convention? Endergonic represent? That is representing what does the bond have of my micromolecules? Do they have energy or do they don't have energy? So when I break down the bond, I'm breaking down the proteins and the energy come out. Like here, when I break down the ATP and the energy exit, the energy comes out. So now there is no energy in the bond. The energy exited the bond. That's exergonic, delta G minus. So delta G minus is exergonic because I'm catabolizing. I'm breaking down the macromolecules. So now the energy comes to me, but the bond has no energy. But when I'm storing, blue, when I'm going from micromolecules to macromolecules, I am building bonds. I am making bonds. So when I'm making bonds, the energy is restored within the bond. That's why we call it plus delta G, endergonic, plus delta G, endergonic. So that convention, exergonic, endergonic, this is not describing energy in my body. That's describing energy in the excellent, in the bonds. So you should tell that catabolism, what's happening in catabolism? I'm breaking down. So when I'm breaking down, is there going to be energy in the bond?
No.
No, the energy is released, but the bond, does it have an energy? It does not, it's broken down. So that's an example of endergonic or exergonic. Exergonic, very good. Now let's say the opposite. I had pancake, or I had steak protein, or I had, the steak protein is a nucleus that has DNA. So those are also nucleotides. So I'm going to assemble those. So when I'm relaxed, I don't need energy. I don't need to break down energy. So now I'm going to form. I'm going to start stacking up. I'm going to start anabolism. I'm going to start building up. I'm going to start storing. So what is that? Is that exergonic or endergonic? Endergonic. Okay. So exergonic, endergonic is no longer complicated because that is just describing the state of the energy within my bond. If there is energy, so that's endergonic, and that's building up. If there is no energy, I'm catabolizing, and the energy left, that's exergonic. Yes. Yes. Usage, yeah. Because the energy can leave the bond. It's gonna leave the bond and come where? It's gonna leave the bond and it's going to come to me. It's gonna leave the bond and it's going to come to me. So this is happening all over. This is happening all over these guys. The amino acids, the nucleotides, the glycerol and the free fatty acids, and the monosaccharides. Okay, so these are amino acids that are building up into, so I gave an example of here, the nucleotide. So nucleotides, they're not just used for DNA. So nucleotides are very useful because they are 2-in-1. They are there for me in the nucleus. So if I want to transcribe some form of mRNA, if I want to replicate the DNA for cell division, so they serve genetic purposes. But that's not the only thing that the nucleotides do. They are like closet hangers. You know the closet, it has many hangers. You put your clothes in there. If you have a lot of clothes, you store it there. But when you want to get dressed and leave, you take it from there. So if you look at the ATP, it's obviously a micromolecule. It's not a macromolecule. Macromolecule, that would be nucleic acid, like the DNA and the RNA. That's a multiple nucleotides. But the ATP is a nucleotide. It's adenine triphosphate, or cytosine triphosphate, or guanine triphosphate. But in the end, all you need to know is that we have a nitrogenous base. These guys are cytosine, guanine, adenine, thymine. These are known as nitrogenous bases. So you have a nitrogenous base like adenine, connected to a sugar that is a ribose sugar. Then that right here, those two, that's a nucleoside. So on the exam I asked you, it is safe to say that the ATP is a nucleoside with 3 phosphates, pi1, pi2, and pi3. So what is the law? The law says if you are 3 out of 3, you are a nucleotide. So if you are a nitrogenous base, a sugar, and at least 1 phosphate, You are a nucleotide. You are a general. You are a general. You are 3 out of 3. 1, 2, 3. Adenine, sugar, phosphate. But if you lose the phosphate, and now you're only 2 out of 3, only adenine and a sugar, that and a ribose sugar is called a ribose sugar, and a ribose sugar, if you're 2 out of 3, then you are nucleoside, and if you are 3 out of 3, you're a nucleotide. So the question on the exam
Yeah, that
was
Yeah, because the answer was nucleotide and 3 phosphates, no?
no. If you say nucleotide and 3 phosphate, okay, please listen. If you say nucleotide and 3 phosphate, we know that ATP is adenine, sugar, and 3 phosphate. What does the law say? It says 2 out of 3 is what? Your name is Lynette. 2 out of 3. What does the law says? Look here. 2 are known as nucleoside. 3, nitrogenous base, sugar, and at least 1. It don't have to be 3. Just 1 is enough. 1 phosphate. That's now the upgrade. That's upgrade from you go from lieutenant to general. So now you're no longer a nucleoside, you're a nucleotide. So the question was saying ATP is a nucleoside with triphosphate or nucleotide with triphosphate. I'm sorry?
Nucleoside?
No.
I misunderstood the question.
Okay, please listen to me. I'm gonna repeat it one more time. Okay, let me repeat it one more time. Okay, let's do this. The law says, please pay attention. The law says that you have to be a nitrogenous base. How many kinds of nitrogenous base do we have? We have adenine, we have guanine, we have cytosine, we have thymine, and we have uracil for RNA. But for now, that does not matter. What matters is we must have one nitrogenous base. In addition to it, we must have a ribose sugar. This ribose sugar, just like that, if you have those two, you already are a nucleoside. Right here, 2, that's a nucleoside. Nitrogenous base and a sugar, that's a nucleoside. If you add 1 phosphate plus pi, it turns into a nucleotide. Okay? So now you can call ATP nucleotide plus 2 phosphate. Because remember, in order to be a nucleotide, you gotta be 3 out of 3. 1, 2, 3. 3 out of 3. So an ATP is a nucleotide with 2 phosphate or a nucleoside with 3 phosphate.
I also made a mistake because I couldn't understand the voice. I know nucleoside and nucleotide, their difference is phosphate, but it was the wording, like meaning that it was a ribose sugar plus plus the adenine, guanine, a nitrogenous base, and a phosphate, or were you saying 2 plus, like the wording was?
Okay. We see now what comprises a nucleoside. What comprises for me a nucleoside? Yeah, and I try any of those. By the way, I don't have to be ATP. I could be GTP. I could be CTP. I could be UTP. I could be GTP. I'm just using that as an example. So on the next exam, I can say it is safe to say that CTP or GTP is a, then I'm going to put nucleoside diphosphate or nucleoside triphosphate. You already know if I am 2 out of 3, I'm a nucleoside. If I have 1 phosphate right here, now I'm a nucleotide. So ATP is a nucleotide with 2 phosphate, or it's a nucleoside with 3 phosphates. Yeah.
That makes sense.
Okay. Okay, no problem, no problem. Let's march so we can cover as much as we can. That is nucleotides. Going from nucleotides to amino acid. So now you see, when I add this phosphate, so now this ATP is charged. The closet is hanging a lot of phosphate. This is storage now. That's anabolism. I built. When I added, it was adenine monophosphate. I can call it adenine mono. Mono is one. Adenine monophosphate is if I only have, is everybody paying attention, please? If I have nitrogenous base, sugar, one phosphate, that is known as adenine monophosphate, because mono is one. I have only one phosphate. I have adenine monophosphate. If I add another phosphate, another PI, that is known as adenine diphosphate. If I add a third one, that's known as, remember, it's all adenine. Adenine the sugar. So adenine monophosphate. Adenine, two of them diphosphate. Adenine triphosphate. So when I built up,
It's starting to close now.
okay, don't worry. All right. Okay, all right. It's about time. Okay. All right, so we have to cover as much as we can. ATP. So now I'm stacking up. I'm building up. So when I'm adding, when I'm putting on the hanger, the three, when I'm putting the last PI here, and I became ATP, going up this direction. What is this? Is this catabolism or anabolism? That's anabolism. Anabolism. Is this storage or breakdown? Storage. I'm storing. Okay. Now. Okay. You're taking time. You're taking time from the very pivotal explanation. And if you go the other way around, now be careful. If you're running the marathon, you're gonna start breaking the bonds. So you're gonna go down from ATP to ADP, and you're gonna get energy out. So energy is gonna come out minus delta G. You go from ADP to AMP, energy come out. Go from AMP, break that bond into a nucleoside. So now the closet is empty. You used all of the phosphate. Phosphate 1, 2, 3. So going in that direction, that's catabolism. That's breakdown. Now there is no, the more I break down, the more energy come out. So there is no energy in the macromolecule. The macromolecule is lacking energy. Minus Gibbs, minus delta G, that is known as exergonic. But if I'm going the other direction, I'm adding to the closet. I put one phosphate, so I go from nucleoside to adenine monophosphate. So I add one phosphate here, so I add energy. So that's plus delta G, that's endergonic. Now I add one more phosphate. Now it's adenine diphosphate. I used to be adenine monophosphate. Now I go up adenine diphosphate, that's even more delta G and more endergonic. Then I add more phosphate, that is delta G, and that is known as more endergonic. So the question on the exam also asked you that this ATP is a form of energy supply because it's a closet. It's supplying me with energy. When I want, I break the bond. I get the phosphates out. So the phosphates come out. So it's the source of energy supply, or is the source of energy storage, or both. What is it? It's both. Because if I have too much energy, I hand P. If I want energy, I take from the closet. Take one P, take one P, take one P. So my ATP is 2 in 1. It's for storing energy and it's for supply energy. Yeah, it's for both. It's for supplying and storing. I'm sorry? It's for both. So we use ATP for catabolism and we use ATP for anabolism. So that is my nucleotide monomer, and that is my nucleic acid polymer. So now let's go back to the amino acid. That was the structure. That was the structure of my okay. That was one form of micromolecule going up to macromolecule. Now let's look at the amino acid. The amino acid look like that. You need to know the structure of your amino acid. It has an alpha carbon. It has a central alpha carbon. It has a central alpha carbon. This alpha carbon has 4 terminals: north, east, south, west. North, east, south, west. North, east, south, west. All of them have 3 out of the 4 in common. All of the amino acids have hydrogen terminal, hydrogen terminal, hydrogen terminal. All of them have on the right. So this is the question that you asked me. What is on the right? What is on the left? So the convention of looking at the amino acid is to look at the left will always be my amino terminal. So this group right here, this is known as the alpha carbonyl. Always to the right, because it's next to the alpha carbon, it's known as the alpha carbonyl, and it has an OH. To the right of the carbon is always known as alpha carbonyl with an OH. Alpha carbonyl with an OH. Or the carboxylic group. But we call it an alpha. This is the alpha carbon, this is the alpha carbonyl because it's next to the alpha carbon. And that's my OH. That's always to the right. Always to the right.
So put it like this, your carboxyl group is always to the right.
Exactly.
Cool. Got it.
And the left is my amino group. So it's N with 3 H's, N with 3 H's, N with 3 H's. It's N with 3 H's. So now all the amino acids have those 3 in common. What makes them different is this R group. And this R group, it makes them different because if that R group is an acid, that makes the amino acid acidic. And if this R group is basic, this is R1. This is R2. This is R2. And this is R3. So it is the R group that tells me if I am acidic, if I am basic, if I am neutral. It is my R group. Yes. The group to the left is called the to the left is the amino group. That's the amino group. That's the carboxyl group. This is the amino group. That's the carboxyl. That's the carboxyl group. That's the amino group.
I have a question.
Yes.
You see how you have O, like O stands for oxygen. The 2 lines means what? Is that the bond?
This is a double bond. So oxygen. So for example, in carbon dioxide, carbon with 2 oxygen. Remember we talked about this. This is an example of what kind of bond? That's an example of that's an example, not polar. Polar is in water. When you have oxygen with 2 H's and the electrons are shared, but they spend more time around the oxygen and just a little bit of time around the hydrogen. It's like when 2 people are sharing the rent or 2 people are sharing a car for a week, but somebody gets the car for 4 days because they're so big and the other person gets it only for 3 days. So that's a polar bond because it's polarizing toward one party. Like here, oxygen is very electronegative and hydrogen is very electropositive. Hydrogen is very electropositive and oxygen is very electronegative. Oxygen is very electronegative. Hydrogen is very electropositive. So now you see there is polarization. This is a polar covalent bond. How do you tell if a bond is covalent? If they're sharing the electron. But if you look at sodium chloride, NaCl, In sodium chloride, sodium gave electrons to the chloride is minus, and sodium is positive. So this is not sharing. Is this sharing? They're not sharing. He gave the electron. That's why he is positively charged, because he lost the electron. And that one is negatively charged because he gained an electron. So he is negatively charged. So now no sharing. But these guys, they are sharing. No one took it than the other. But the sharing is not fair, is not equal, is polarized. So here the oxygen is very electronegative, while the hydrogen is electropositive because the two electrons that are supposed to be shared is actually like this. They're closer to oxygen, than they are, even though it's supposed to be equal. It's supposed to be equal. So they are covalent, they are sharing. No, the oxygen didn't take it yet, but they are polarizing toward the oxygen some more. But in carbon dioxide is equal. So 50% of the, so these are two electrons. So two of the electrons are circulating around carbon, and two of the electrons are circulating around oxygen. Two of the electrons are circulating around carbon, and two of the electrons are circulating around oxygen. So this is 50-50. That is non-polar. So what does polar mean? Polar is when you're more tilted to one side than the other. Polar doesn't mean you're more cool. It just means that the relationship is unfair. The electrons are polarized to the oxygen more than they are. But here, they're equal. So the double bond, we're sharing electron. We're sharing electrons. We're sharing electrons between the oxygen and the carbon. So that's an example of what kind of bond is that? The oxygen and the carbon. Well, the oxygen and carbon, what kind of bond? Please, hold on. Yeah. So now look, if we say what kind of bond, ask yourself a question. Are we sharing, or did one give the electrons to the other? So if you look at this, so in sodium chloride, one is electropositive, one electronegative because he lost. So they have a charge. They can have a charge. So whenever there is a positive charge, somebody lost an electron. Whenever the negative charge, somebody gained an electron. Here, do you see a charge? So that means nobody gave nobody nothing. Nobody accepted anything, because if they accepted, they would be negative. Because we don't, it's not like the bank. When you get money, you get plus $100. The currency of the bond is electron, is minus. So actually you get minus. If you get 10 minus, it's like you got, it's not $10, it's 10 electrons. So you get paid in minus. So this guy got paid an electron, and this guy lost. That's why he is positive. That's why it's confusing to us. It's counterintuitive, because for us positive is gain. But the language of chemistry is the electron. When you're negative, you gained an electron. When you're positive, you lost an electron. Yeah. So if you want to tell if the bond is covalent or not, is there a charge? There is no charge, so they're sharing. So now this is a covalent bond. If it's sharing, that's a covalent bond. Can you just give me 5 minutes? I'll be right back. 5 minutes, I'll be right back.
But what about the most he's ever actually explained and taught, bro? Like, nah, bro, it took me to move up in the black. Like, it's crazy. Bro.
Assumption.
Like, bro, this is crazy, bro. We finally have a teacher. My whole thing is I pay money for it, so you want to teach, bro. I don't care how I got to say it, whatever you want to teach. You know what
What brought your in a one eye word, yeah. Oh, so
Yes, I guess to make it.
I told you, like,
And when it's outside, that's all right. Transition.
Yeah, we gonna. I'm way on the study the last. Every night I'm on that. Bro.
Yeah. But the equal sign of the time, you know.
You see how we all like, oh.
Yeah, for me, it's on. That's all I read.
I've been away.
Yeah. On the left.
I cannot have it.
Look at it the way it is.
It's on the right.
Yeah. You're looking at it like.
Just read it as it is.
Yeah. Some people are buying it where it's like left is right.
Everything.
They're making, and some of them, they don't know everything.
I'm not.
One thing I will say is like still trying to determine like,
Yeah.
for example, what it is. Yeah, that's the only thing I'm trying to do.
Right. Double bond is for box.
Yeah. Basic or.
You said, so the amino is next, right?
Yeah, more basic.
This one, the carboxy, I know, we put right, it's always like,
And also, the middle part, always in the part.
You know, like how you said, right now, what is a monitor?
appreciate it. What is that? Like, I understand so well.
So you are. That's the important.
I try to do it, but I'm getting like, don't ask me. They don't talk in there. So don't go with all your, you know, the pitiful woman.
You can do this one.
Oh, my God.
Okay.
Pick one like you were doing.
Yeah, so I'm
Like the whole thing, like, no, they're not gonna get all of them.
Digital happen. In the middle of the, but it could be up or down.
Yeah, anytime you want to use the bathroom, you're welcome to go out. You don't have to ask me. You just go to the bathroom. Okay, all right, so let's continue, please. All right, so now we are able to tell that the relationship between two atoms is based on the electron. So the electrons are spinning on the outside, and it is actually the ones who are farthest away from the core, from the nucleus. These are the ones who get shared or they get given off like an ionic bond. So you have to know the ionic bonding because that's we're going to learn how we do the folding. So when we go from a primary structure, protein, to a secondary structure, to a tertiary structure, to a quaternary structure, that's through the ionic bonding. So when you heat up the protein, why does it disintegrate and fall apart? Because the ionic bonds are broken. Because the hydrogen bonds are broken. Okay, because the hydrogen bonds, so you guys remember the hydrogen bonds are kinds of bonds that result like dotted bond, like a weak bond. For example, they only happen, so that's why they call them hydrogen bond, because they are always between hydrogen and some other electronegative atom, usually oxygen or nitrogen. For example, or chloride, but it, or fluorine. Okay. Yeah, but most likely that is happening in that is happening most of the time in the amino acids. They don't have fluorine, the amino acids, and that's happening in water. We're so concerned with water for solvation, for solutions. So if you look at water, water looks like this. O with two H's, O with two H's, O with two H's, O with two H's. So this bond between the O and the two H's, what kind of bond is that? That's no. So before we specify, is it polar, covalent, or non-polar? Begin big. Begin the continent, North America, the country, America, is not Canada, the state, New York, then the borough. So begin far. When I say what kind of bond, first identify. Is it covalent? Is it ionic? Is it hydrogen bonding? Or is it van der Waals? So this bond here between the O and the H, is what kind of bond is that? No, that's covalent. Remember I told you, if you want to tell what kind of bond is it, ask yourself one question. Are the electrons shared or are they not shared? How do you tell if they are shared? Everybody's neutral, because like we did with this, carbon dioxide. There's no charge, so everything is shared. Water has oxygen and hydrogen, they are shared. Yes, it is non-polar, so the electrons are polarized. That's why we put this symbol that this is more magnet, this is more electronegative. This is electronegative, and that is electropositive. But that's not a charge. That's not a charge. That's just a direction, a polarity of who's more electronegative. But here, this bond, what kind of bond is that? That's no. This bond, what kind of bond is that? That's covalent. So when I ask what kind of bond is this? Ask yourself. You look at the periodic table. Ask yourself, is there sharing, or did one get the electron and the other lost the electron? There's only way to find out if one got the electrons and the other lost by a chart. If you see a chart, then you know somebody lost, somebody gained. Who lost? The one that has positive. Who gained? The one who got paid in minus. So now when you look at this, do you see a chart? No. No chart. Then that's definitely covalent. Okay. In covalent, you're not done. New York, now you got to find the borough. Is it polar covalent or non-polar covalent? So how do you know? Excellent. How do you know if it's polar covalent or non-polar covalent? Polar covalent usually happens with a huge, very polar molecule, like oxygen, like nitrogen, like fluorine, who's bonding with a really small. That's why you're not supposed to have a contract with a minor, because there's an adult and a minor. So that's exactly what's happening here. These are having a contract. The oxygen is a big adult, and the hydrogen is just it's called a hydrogen proton. It doesn't even have a neutron. It just has a P plus. So they're having a relationship now. So that's why it's non-polar, because all the electrons are actually more toward the oxygen. So it's covalent though, but it is polar. It's polarizing to the oxygen. That's why the oxygen is more electronegative, because the electrons were supposed to be shared equally, like here, they're actually more towards him. But when oxygen is in relationship with carbon, is not the electrons are not closer to oxygen, they are equal. So that is non-polar. They are not polarizing. Here, they're polarizing. So in water, this bond, H2O, what kind of bond is that? Covalent. Is it polar or non-polar? So always don't say that's polar. Begin with covalent or non-covalent. Then polar or non-polar. So this bond, what is that?
Is it, hold on, one second, is it covalent because it's sharing with both H's?
It's covalent because it doesn't have to be two, just one. If you're sharing a bond like this, that's covalent. You don't have a charge. Does the oxygen have a charge? It doesn't, so it's covalent. It has another covalent bond, that's something else. But as long as no charge,
Covalent,
that is covalent
all
bond,
the time.
all the time.
You will always show a charge?
Of course, if the relation, if the amino acid, they have a minus charge, so we will talk more about that. But we're not really gonna get tested about that. Just remember that this is a covalent bond, and this covalent bond, so that's not if you have water, these are not the only bonds that you have. Because all the oxygen, so this oxygen is more electronegative because he's so big, and these are weak electropositive. This electronegative, and these are weak electropositive. So do you think that's all that's happening in the water solution? No. The oxygen is now gonna start having a green bond with the positive electropositive hydrogen of the other oxygen. And this oxygen is gonna start to have a weak green bond with this hydrogen. And this hydrogen is gonna have a weak bond with this oxygen. So there are two types of bond. This is known as an intermolecular bond that is covalent. It's covalent, but it's intermolecular. This is, I'm sorry, it's intramolecular, within. It's within the molecule. It's intramolecular. Within the molecule, intramolecular. The green bond, that is intermolecular, like international. International between two nations. The US and Jamaica, that's international. So these are three molecules. The green bond is intermolecular between, these are three molecules. One, molecule two, molecule three. So the green bond is connecting the three different molecules. So that's intermolecular. That is intermolecular bond. That's an intermolecular bond. But the bond that is inside the molecule, that is not inter, that is intra. That is intramolecular bond. So the intramolecular bond is covalent. That's covalent polar. That's a polar covalent bond, the red one. The red one is a polar covalent bond. This one, that's a polar covalent intramolecular bond. And the green one is between two molecules. So that is intermolecular, international, between two nations, the US, Jamaica. So that is intermolecular bond, and it is a hydrogen bond because it's between the positive, electropositive hydrogen of one molecule and electronegative oxygen of the second molecule. So that is intermolecular bond, intermolecular bond. So that's a hydrogen bond. So that is how the proteins fold. So proteins, these amino acids, you see the amino acids, they look like that. The amino acids, that's their structure. Let me go back to the structure of the amino acids. The amino acids have a alpha carbon. The amino acids have an alpha carbon, amino terminal, and a hydrogen terminal. Alpha terminal, an amino terminal, and a hydrogen terminal. So I'm going to show you how these hydrogen bonds are responsible for the folding of my amino acid. So now at least you know what's the difference between intramolecular and intermolecular. Because the folding of the proteins from a primary structure to a secondary structure like alpha helix and beta sheet, like alpha helix and beta sheet, these are types of hydrogen bonds. So always remember your amino acids. Amino acids are alpha carbon. Please say the amino acid with me. Alpha carbon. There is like a alpha carbon here. Alpha carbon. The second one. Alpha carbon. Alpha carbon. Alpha carbon. Those are 3 alpha carbon. Alpha carbon. Okay. They have 3 equal. 3 same terminals. Hydrogen, 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 hydrogen. And they have an alpha carbonyl on the right with an OH, that's acidic. With an OH, that's acidic. With an OH, that's acidic. OH, that's acidic. Then on the left has an amino with 3 H's. 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 And then you lastly have your R group. R1, R2, R3, R4. These are our 4 different amino acids. Now, all of these bonds are what kind of bond? All of these bonds, the blue one, the red one, are they inter or intra? Look, please look, please look. When you say international, what does international mean?
Between.
An international flight.
Between New York and Paris.
So international is when you say between New York and Paris, these are 2 cities. But international is between 2, yes, you go to the country through the city, but international is between 2 nations, 2 separate nations. America, Jamaica, America, Canada, America, Mexico, international. But when you go from New York to Tennessee, you're still inside the same nation. That's intra. So intra-domestic, thank you. Intra is inside, within, within. Intra is within. Intra is inside, within. Inter is between 2 molecules. Like the water, it was like that. That's water molecule number 1. That's water molecule number 2, H2O, right? These are 2 molecules. The blue bond, these are intra, inside, within, intra, inside. But the green bond, because this one is more electronegative, and these guys are electropositive, they're gonna start flirting, they're gonna start having flirting relationship. That is
Inter.
Okay. Thank you. So the intermolecular is between 2 sovereign, 2 autonomous different molecules. Intra is inside. The blue one is intra. The green one is inter. So that is an example of a hydrogen bond. The green one is hydrogen bond because it's hydrogen flirting weakly with the more electronegative oxygen. But the blue one is not weak, it's strong. That's covalent, that's sharing. They are sharing electrons. Yes, they are polar because more toward this guy is more electronegative. That's why we even have hydrogen bonding, but it is covalent. So when you look at the amino acid, you gotta know the structure of the amino acid. That alpha carbon. On the right of it is your alpha carbonyl. It has an OH. That's the acidic side. It has N with 3 H's. That's an amino side. And they cancel each other. Then you have the R group. That is what gives the amino acid its identity. You must remember that the amino acid is a buffer system. So when your pH is high, what does that mean when your pH is high? It means that this was another question on the exam. That means the hydrogen proton concentration is low. That means the acidity is low. That means the basicity, basic is
High.
high. That means the hydroxyl concentration is high, because when you are basic, your hydroxyl is high. That means the alkalosis is high. That's high. So these are all the same. So alkalosis is high, meaning pH is high, meaning hydrogen proton is low, the acidity is going to be low, basicity is going to be high, hydroxyl concentration is high. If you have acidosis, then if the acidosis is high, that means the pH is going to be low. That means the hydrogen proton concentration is going to be high. That means the acidity is going to be the acidosis. High hydrogen proton. The acidity is high. Excellent. That means the basic is going to be low. That means the hydroxyl concentration is the hydroxyl concentration is going to be low. And what about 2,3-BPG? Due to 3-BPG is a metabolic waste. Metabolic waste. So it's going to be high. Metabolic waste is going to increase the acidity. Metabolic waste high. The metabolic waste is going to be high. Metabolic waste is going to be high. For alkalosis, metabolic waste is going to be metabolic waste known as 2,3-BPG is going to be low. And what about carbon dioxide? What about carbon dioxide? Carbon dioxide is like acid. That's why when you have acidosis, your respiratory system goes, you're hyperventilating because you're shunting carbon dioxide, carbonic acid. So carbon dioxide is going to be high for acidosis, which is the same thing as carbonic acid. Carbonic acid look like this. H2CO3. That's going to dissociate in blood to give you CO2 plus H2O. And this is known as carbonic acid. So when I ask you on the exam, what are the features of a toxic metabolic waste suffocating tissue that need hemoglobin? It's this one. The carbon dioxide is high. The metabolic waste is high. The 2,3-BPG is high. The acidosis is high. The pH is low. Hydrogen proton is high. The acidity is high. The basic is low. The hydroxyl is low. These are all the same. It sounds a lot, but it's the same. Because if the basicity is high, acidity is low. If the hydrogen proton is high, the hydroxyl is going to be low. So I need you to look at it in both directions. They sound long, but if you get one, you know the other. If it's sunny in one part in the North Pole, it's dark in the South Pole. So it's just two different things. So don't get thrown off by a lot of information. But it is a reflection. I'm just mentioning the reflection. So a couple can be able to guide you to the others. Yes?
So then when it comes to the T state and R state, 2,3-BPG or whatever, is that attaching to the T state?
So it is not okay. The T state and the R state, what are they? These are description of who? Of hemoglobin. So for example, I'm describing the state of hemoglobin. Is it full of oxygen? That's R. If it's not full of oxygen, it's T. If hemoglobin is full of oxygen, it looks like circles like that. Now that is R state. It's full of oxygen. If the hemoglobin is not full of oxygen, is all squares, then it is in the T state. So the people who smoke cigarette and the people who have carbon monoxide poisoning, Carbon monoxide is gonna competitively bind to the location that oxygen was supposed to bind to. That heme group, that seat, that VIP seat that the oxygen is supposed to be sitting on in the R state is going to go to the tissue. Now hemoglobin is carrying carbon monoxide poison, is delivering poison now. So carbon, but we cannot call that an R state because the R state has to be oxygen fully saturating hemoglobin. But carbon monoxide prevents that. So it stabilizes the T state, the enemy, takes no oxygen. So carbon monoxide, no oxygen. Carbon monoxide, T always. It is the oxygen that's in the R state. So if you have emphysema, if you smoke nicotine, these are the guys. So the question was saying, stabilized in the T state, stabilized in the R state. That's just telling you if you have nicotine in your blood, if you have carbon monoxide, what is the shape of your hemoglobin? Is it full of oxygen or is it empty with oxygen? So with carbon monoxide, yes.
So, all right, so just to piggyback on what you said, I feel like if you would have like rephrased that question a different way, because like some of your questions were like, didn't really understand what you had.
I'll do that. I'll do that. So I'll take what your friend or what you guys said. I'm gonna spread out like one question, break it into two easier questions.
Right.
But few ones, I'll compact them, but you can get them. You can get them. Just like for example, this one. This one compressed a lot of stuff, but it's actually the same. If you think about it, if alkalosis is high, you know the pH is high, you know the hydrogen proton is low, the acidity is low, the basicity is high, hydroxyl is high. So yeah, don't worry. Let's now learn. I'll make a nice review session for you, and I'll make sure that we all march and we do well on Wednesday. Yes.
Another thing, there was a question on the quiz. I don't remember like specifically, but long story short, basically it's like how many like you see how you have that in red, like the equation on top. It was like how many like bonds do you see or something like that, like
Peptide bond.
Yeah,
Okay, so
yeah,
I'm gonna
yeah.
show you this one now. So it's very simple. I'm gonna show it to you. So look at this. So now we're looking at the structure. So you have to know the structure of your micro and your macromolecule. So you have to know the structure of your micro and your macromolecules. We talked about the structure of our nucleotides. They have to have 3 out of 3 if they are nucleotide, nitrogenous base, and sugar and a phosphate. Phosphate is the third one. If there's no phosphate, 2 out of 3, that's a nucleoside. Very good. Okay, so now when we look at this, when we look at that, you have to understand, if I am building up, if I am building up, that is anabolism. So when I'm building up anabolism, I make big macromolecule like protein, I make big one protein going from small one amino acid. Same thing for the nucleotide. I go from nucleotide into nucleic acid. If I'm going up, that's anabolism. How do I go up? I go up by removing water minus H2O. That is known as dehydration. I remove water. So I remove which water? So now I know my structure already. I have alpha carbon to the rightmost. This side is my alpha carbonyl with its OH. To the leftmost, that's your amino terminal. So these are separate. These guys are separate. That's why we call it R4, because that's amino acid number 4, amino acid number 3, number 2, number 1. So if I am building up, I'm removing minus water. So how am I gonna get minus water? So those two have to be bonded, right? They're gonna get married, they're gonna get bonded. So who's gonna give the OH is the carboxylic terminal. The carboxylic terminal is gonna lose the OH. And the amino terminal is gonna give the H. And now water gone out. And now a bond is made. That's a peptide bond. And now that's anabolism. Now these are not amino acid no more. That's a couple now. They're a couple. Again, the carboxylic terminal is gonna say I pay OH. And this amino gonna say I give an H. And then water minus water went out and the bond has been made. So now there is energy in the bond. Is that catabolism or anabolism? Is that exergonic or endergonic? And I'm building that. Now energy is being made. Look at this now. OH and an H. So this one is gonna give an OH. This one is gonna give an H. And a bond is gonna be made. That is known as peptide bond. That's now I no longer, they are not separated. They are not separated, they're all attached. So that's a primary structure.
So just to make sure, I'm looking at 3 peptide bonds right now.
Yes,
Okay.
yes, 3 peptide bonds. 3 peptide bonds always between these guys. Because who gets married? The carbonyl, and the carbonyl gives an OH. And the amino gives one H. And then they have the bond. So on that structure, I'm gonna put up the structure on the board. Look for this. That is a peptide bond. That is a peptide bond. A bond between N and 2 H's. Because remember, it used to be N with 3 H's. And N with 2 H's and a carbonyl that lost an OH. Yes.
Carbons, like I was asking down.
Without the carbon. So the carbon can be in a so there are different configurations of displaying the macromolecule. If you see a dot, I will go over it now. If you see a dot, that's a carbon. So when we're doing the fatty acid, we said the fatty acid looks like this carbon with OH, and that's a carbon, that's a carbon, that's a carbon, that's a carbon, that's a carbon. And you have glycerol, that's OH with carbon, with carbon, with carbon, OH and OH. And what happens, the same thing. This one gives an OH. I'm sorry, this one gives an always the carbonyl gives the OH, and this one gives an H, and there's a bond like that. That's the triglycerol. If you want to rewrite it, you go like this. You can remove the carbon, put a dot like that. Carbon, put a dot like this. Carbon, put a dot like this. Carbon, put a dot like this. So now that it looks like, no, that's a carbon. So whenever there's edge, that's a carbon. When there's a dent, when it's dented at the corner, that's a carbon.
Is that what that means?
So the amino group is different. The amino group is right here. So here, that's not a dent because at the end of the dent, there's a nitrogen. But if there was nothing at the end, then like here, now these are 2 carbons. But we don't write them. Like that, you already know these are 2 carbons. So always look, you know, when I connect 2 amino acids, don't get fooled by there is a big aromatic group, there is a cyclic group. Forget about that. Look for the N and the alpha carbonyl, even if they have no structure of alpha carbonyl. So you just look for N and a carbon that has a 2 oxygen bond, that's a carbonyl. That's called a carbonyl. So this carbonyl has a bond between the nitrogen, between its carbon and the nitrogen. So that's 1 peptide bond, that's 2 peptide bond, that's 3 peptide bonds.
So just to simplify, I want to make sure I understand you. So what you're saying is, if you have a bond between C and N, that's what makes it a peptide bond.
No, they're gonna trick you. They're gonna put a bond between some, like this C and N. Is that a peptide bond now? The C has to have the carbonyl. You know that's your alpha carbonyl. You know that one. That's the alpha carbonyl that used to have an OH, and she gave it off. And the other amino gave off 1 hydrogen, and now you have H2O gone. So you have to remember that, even though it's not there, you visualize when I go up from amino acid to protein, I remove water.
said, so it has to have the carbon on it, for example, the question in the
Mhm.
exam, where you showed the peptide bond, and the answer was 2.
So it has to look like this. The carbonyl and a nitrogen with the carbon. So it has to look like that. Nitrogen with two H's. Okay, can you pay attention, please? Okay, so just show them later. For now, please pay attention to here. If you have to be able to remember that going from amino acid to protein, I minus H2O. And if I want to break down, I add H2O. I replace it right back. If I want to replace this bond and say, hey, remember back in the day you gave us an OH? Here you go. That's your OH back. You had an H, we divorced. So now you take your H, you take your OH. You want to rebond. Okay, please give me an OH, please give me an H, water comes out. So you should remember that this bond was not made for free. It was made because somebody gave me an OH and somebody gave me an H. Who gave me my OH? The carboxyl group. Who gave me my H? My amino group of the other amino acid. That's how they're bonded. So you're looking for that structure. You're looking, so look for it in the review session. I'm going to go over every single question. So look here, the carbonyl and the nitrogen, they are connected. So that's a peptide bond. That's a peptide bond. So what I want you to understand is that when we're building up, when we're storing, when we're anabolizing, we remove water minus H2O. That was on the exam. Dehydration synthesis. And you saw that one gave an OH and one gave an H. That's H2O. One gave an OH, one gave an H. That is an H2O. And we removed everything. Yes.
And all right, this discussion was asked,
So you see that's a carbonyl, and that's an N with two H's. So this is N with two H's. This is an N with an H. Okay, so this is
but the question,
Process of bringing water
the
inside
question
us,
was asked,
and adding water back.
what amino acid does it have is it with an ion?
yeah. I'm gonna go over that. I'm gonna go over that. Don't worry. I'm gonna go over. I'm gonna go over every single question on the exam on that recorded Zoom. So just listen to it. You will get all the answers. Okay. This, remember, when I am adding things up, it is no longer a puzzle. When I'm adding things up, I remove water, minus H2O. When I'm splitting, I add water. Put water back. Here you go. That's your OH. That's your H. Add them up. Remove water, minus water. Peptide bond. So you have to know, we add by removing water. Dehydration, removal, devoiding.
So basically making OH separate from H. You know?
Yeah. So making an OH is not making OH separate from H. You either make water or put back water. So these guys used to look like this, right? OH and H. They are separated now. If I want to join them, make a peptide bond. What I do?
You
That's 360.
double bonded and you take one away. Yo, you take away the H. Take away the H.
You take the H and the OH. So now you remove H2O. You remove both of them and you connect like that. You connect like this. That's if you want to go up, build up, stack them up. That's known as dehydration. But if you want to cleave the bond, that is now hydrolysis. Lysis is break. Hydrolysis. Okay, so breaking that. So this is my amino acid. So now these are my peptide bonds. So there was a question also. These are my peptide bonds. These are my peptide bonds. My question is that when I get this long linear chain with connected peptide bonds, long linear chains with connected peptide bonds. This is now a polymer or a monomer.
Polymer.
The polymer is connected like a train. It's connected. When it was not connected, it was monomers separated. Now they're gang together. They're connected. So in a ribosome, so these guys are like a bead. So what happens is in the DNA, what happens in the DNA? What happens, the DNA is my template. So in my nucleus, I have DNA. And the DNA is like that. They did an experiment. In the experiment, there was a nucleus in the skeletal muscle cell. Skeletal muscle cell nucleus. Skeletal muscle cell, and it has a nucleus. You cannot be a cell if you have no nucleus. Then they got an ovum, that's the egg cell, ovum. That's the one that gets fertilized with the sperm. And they remove the ovum, they remove the nucleus of the ovum. And they took out the nucleus of the skeletal muscle of the frog. This is frog skeletal muscle cell, and this is frog ovum. They removed the skeletal muscle nucleus and they planted it inside the ovum. And they brought a sperm. And they said that, we remove the nucleus, and we bring a sperm. I mean, nothing will happen because, that an ovum, it has no nucleus, and now that's the nucleus of a skeletal muscle. When they added the sperm, guess what happened?
It finds a way.
I'm sorry?
Life finds a way.
Yeah, everything went perfect. The union happened between the sperm and egg, then the morula, then the blastula, then the gastrulation, and the gastrulation, and all of this happened until you had an organism.
That could pretty much be stem cell, right? No?
No, it's not a stem. A stem cell is a cell that has the potential to developing into multi-cell. No, that's not what happened.
No?
What happened was that they realized that it is not. You see, these are two different cells. That's a skeletal muscle cell. That's a frog cell. They took the nucleus out from the ovum and they replaced it with the nucleus from the skeletal muscle, and everything went perfect. They discovered that the DNA is universal. It's like you give $1,000 to a police officer, you give $1,000 to an athlete, you give $1,000 to a chef. The chef is going to take the $1,000, buy utensils. For example, the athlete is going to take the $1,000 and buy sneakers and basketball and whatever. The police officer is going to take the money and buy, for example, guns and weapons and taser. So the $1,000 is the same, but how did you use it? So the DNA is the same. Frog cell, neuron cell, muscle cell, liver cell, heart cell is the same DNA. It is the system, the host of the enzymes that transcribe. Remember the central dogma of biology. In the central dogma of biology, this is very important. In the central dogma of biology, what happened was we have DNA and we transcribe it into RNA, mRNA, rRNA, and tRNA. These are different forms, but still RNA. And then we go from that to making a protein. It is the protein inside the skeletal muscle that makes it different than the heart muscle because the proteins will make you different. So if you compare the proteins inside the heart cell, they are cytoskeletal proteins because they have to pump a lot of blood. They have to be pushing a lot of blood. But if you look at the proteins of the neuron, there's a lot of protein channels to allow the entry of electrolytes and chain membrane voltage. So you see, it's the proteins who make the cell different from the other one. But how do we get proteins? From DNA. We transcribe. We transcribe. We transcribe. We transcribe. Transcribe DNA to get mRNA, and we translate mRNA to get a protein. So if I want to become a skeletal muscle cell, I prescribe a region here known as the skeletal muscle protein. If I transcribe that, I'm going to get mRNA that will give me skeletal muscle protein. If I'm a neuron, I go to a separate region, the neuron, and I transcribe the DNA to get neuron mRNA to get neuron proteins. If I want ovum, if I want to become an egg or a sperm, there is a separate region. The ovum is here. So the DNA is going to be DNA region that has for the ovum is going to be transcribed, not the skeletal, not the neuron. For the ovum is going to be transcribed. Then the RNA is going to be translated to give me ovum protein. So they realized even though the nucleus is not the same, but the DNA is the same. It is my polymerase. My RNA polymerases, these are the ones who target where to go. No, we are the ovum. We polymerize proteins that help an ovum become an ovum. We help the police officer become a police officer. We help an athlete become an athlete. We help a chef become a chef. So it is the set of polymerases. They will target a region in your DNA and then they will transcribe it. That's the key thing. So just because you have a DNA in the cell, it does not mean it's all active. It is the region known as the euchromatin that is when transcribed will give you. So this now will make sense. Let's see the polymerases. So obviously we have to have some terminology. What's a polymerase? People get confused with DNA polymerase, RNA polymerase, DNA polymerase, RNA polymerase. People get confused with that. Just break down the word. Polymerase. What's an ASE? Enzyme. So if you're an ASE, if you're a polymerase, what do? You make polymers. So that means you are anabolic or catabolic. Anabolic, because you're making. So if you make a polymer of DNA, if you make a DNA polymer, then you are a DNA polymerase because you make a polymer. So if I have a DNA polymer, who made that for me? Impossible. RNA polymerase. Impossible. Because RNA polymerase makes what kind of polymer? RNA. Excellent. That makes for me an RNA polymer. So if I want to make RNA, I promise you, the one who will be used is going to give me an RNA polymerase. To get an RNA polymerase, to get an RNA polymer, I'm going to use an RNA polymerase. So this is an enzyme that polymerizes, put beads on the string. It doesn't remove the beads. It makes big. It's going up. But the issue now is so here, my DNA, I transcribe the DNA to get RNA. So who am I going to use now when I transcribe the DNA to get mRNA? What kind of polymerase am I going to use to get mRNA, to get here, to go from DNA to mRNA? So what polymerase am I going to use? RNA. Excellent. So I'm going to use RNA because I'm going down that direction. I'm going to use RNA polymerase. RNA polymerase. Who did I depend on? Who did I read and transcribe from? DNA. That's why we call it DNA dependent RNA polymerase. But don't forget the HIV virus is a retrovirus because it goes the other way around. This is the central dogma of biology. But the virus does not listen to the central dogma of biology. The virus has viral mRNA, and it has this very wicked enzyme known as reverse transcriptase. You see always A's, everywhere A's, because these are enzymes. So it's a form of polymerase too, because it polymerizes to give me what? DNA. So reverse transcriptase is what kind of polymerase? It's a DNA polymerase because it polymerizes DNA. So now who did it depend on? The RNA. So that's an example of a RNA dependent. Excellent. DNA polymerase. How did I know that? Because it makes for me a polymer of DNA from the mRNA. And then it will hijack your ribosome to translate. It will hijack the human ribosome to translate for me this viral so we went from mRNA to viral DNA, then it will hijack the cell polymerase in order to transcribe the DNA of the virus into viral mRNA, into viral proteins that will kill the cell. So that's an example of an RNA-dependent DNA polymerase. In replication, yes.
And this only happens in HIV or someone?
Yeah, this is just in HIV for now. It's just in HIV. But this is to show you that I polymerize different ways. But in the end, break it down. Am I polymerizing? That's polymerase. Polymerizing what? DNA? That's DNA polymerase. Polymerizing RNA? That's RNA polymerase. What did I make the polymer of? RNA, then I'm RNA dependent. DNA, then I'm DNA dependent. So let's look at the replication. The replication I would have for my cell division. For the cell division, I have one cell that has to divide. Why the cell gotta divide? The cell has to divide because growth and development, you have a toddler, you have an infant, you have a toddler, you have an infant. That infant has to grow up, become a child, and then an adolescent, then a young adult, then an adult, then like that. So for growth and development, we need to divide the cell. For healing, I'm injured. So the tissue that's dead, the necrotic tissue must be removed by the immune cells and pave the path for the growth of the surrounding tissue to replace that tissue by regeneration and new growth. So this is now the cell cycle. In order for me to divide, the cell has to go through two phases, two big phases that are divided into subphases. The first big phase is known as interphase. This interphase is divided into the G1 phase and the S phase and the G2 phase. This is all called, this here is called interphase. That half is called interphase. That half is called interphase. That half is called interphase. The second half, now I'm going to divide. That's known as mitosis. That's known as mitosis. The first half is known as prophase, then prometaphase, then you have metaphase, then you have anaphase, then you have telophase, then you have cytokinase. That's mitosis. So in replication happens here in the S phase, where I double my DNA. How am I gonna double to another cell? How am I going to double into two cells? So for example, I need to divide for here. Why do I have the cell? Why do I have the cell division cycle? Why do I have the cell division cycle. I have the cell division cycle for growth and development, growth and development. And I have also for healing, So in order for that to happen, I must be able to divide. So if I want to divide, that's my DNA, 1x. I have DNA. And we said that my DNA is double-stranded. My DNA is double-stranded like that. So in order for me to double, I have to double my DNA. So now within the same cell, I'm going to have to double the DNA. So I'm going to unwind the zipper, unwind the DNA into two separate strands. Then I'm going to get, what kind of polymerase am I going to get? To get a second strand of DNA. So I'm DNA polymerase. That DNA polymerase is dependent on what? DNA. So that's a DNA dependent DNA polymerase. That's a DNA dependent DNA dependent polymer DNA dependent DNA polymerase. So what happens, it's going to make a copy of this strand and it's going to make a copy of this strand that is cut up. And this is where your Okazaki fragment, I'm going to talk about those in a little. So they're going to be cut up like that, and then they're going to be ligated and connected by this yellow ligase. So now I have doubled my DNA. So I used to have, these are my original, these are my parent. These are my parental strands. These are my parental strands. These are my parent, the blue ones are my parent strand. These are my parental strands. The purple one and the green one, these are my daughter strands. So now this is 2x, 2x. I used to be 1x, but I doubled everything. Now it's 2x. Are we clear? That's a replication. Replication. I used a DNA-dependent DNA polymerase. I unwind, I cut up the two helix, then I copied, I made a copy and paste, I copied each strand. So now I used to have 1x DNA, now I have double 2x DNA. Then I'm gonna go into my mitosis, and I'm going to now divide into two cells, my two original strands, and my daughter strand, and then both of them are going to separate, and now I'm going to have and now I have 1x of each. So you see I started with 1x. I performed replication with the help of, replication.
DNA dependent, DNA polymers.
Yeah, polymerase.
Yeah, there you go. Yeah.
So I have now, I used DNA dependent DNA polymerase, and it doubled for me the copies. And then I did mitosis. Now I have two cells, and now that's my original cell. It broke into more cells to make up for the tissue that is healing, the tissue that is lost. So this is replication. So what I want you to understand for the exam is I'm going to ask you the reverse transcriptase of the virus. Is it DNA dependent, RNA polymerase, RNA dependent, DNA polymerase, like that? Then I'm gonna ask you. So for the reverse transcriptase, is going to make a DNA, so it's a DNA polymerase dependent on RNA, so that's a RNA dependent DNA polymerase. When I'm transcribing, trying to make an mRNA to give me the protein that I need, I'm going from DNA to RNA, so that's the RNA polymerase. It depends on who? It depends on DNA, so that's DNA dependent RNA polymerase. And for replication, I'm spinning in circle. I double up my own DNA from DNA to DNA. So that's DNA dependent who made DNA. So that's DNA dependent DNA polymerase. Those three are on the exam. So now you got 3 out of 100.
But this is on for the next exam?
Yeah,
The one that we just took and took over? Or this is-
yeah. So it was on the exam, right? It was on the exam.
Yeah, it was.
The DNA dependent RNA polymerase. So these are 3 points.
Not dependent, but. Yeah, the recombinant.
Yeah. So that's the recombinant DNA. So I'm going to just ask you those. Okay, how about we make it 50 questions? We make it 50 questions? Alright. So we make it 50 questions. So far, so what I've told you about today, keep it in mind. And those 3 questions, very simple. If you're making RNA, you use RNA polymerase.
Right.
And that happens in transcription. I want to make collagen. Collagen is a protein. I want to make keratin. Keratin is a protein. How am I going to make keratin if I don't have mRNA? So I need DNA dependent RNA polymerase to give me the mRNA transcript. Then the mRNA is going to go to the ribosome and get translated to give me my collagen. So that's what happened in the frog experiment. When they removed that nucleus, the polymerase is there saying it doesn't matter what nucleus is there. We know where to go. We know which region of the chromosome to transcribe. If they take that nucleus from the ovum and they go put it in the retina of the eye, the polymerase is there saying it don't matter. As long as the DNA is there, don't matter. We know which part that is our proteins, the retinal proteins. The nerve cells will say, give me any nucleus, no problem. I know which part of my DNA to go to do my DNA dependent RNA polymerase and polymerize my mRNA, and then my ribosome will translate for me. So that's the very significant experiment that got them the Nobel Prize, because they proved that the DNA is the same everywhere, and it is those set of enzymes, those set of proteins, they are the ones who are able to go and target which region to go. Yes.
Is that why they wanted to do the human genome? So for this reason?
Yeah, so they did the human genome to see, for example, which regions of the genome are always diseased. Like, for example, sickle cell anemia, that's a mutation. So they know that region always gets mutated, giving me sickle cell anemia. So that's called genomic medicine. We focus on certain parts of the DNA to study them so we can see why this region all the time mutation. So that's why they did that. So you got to know what replication is. Replication is when I double my DNA. What do I use for that? What kind of polymerase I use for that?
The DNA dependent DNA
Polymerase, yeah.
Yeah.
And if I am going as a retrovirus, I have no DNA, I just have mRNA and my reverse transcriptase. What is reverse transcriptase?
In order to use that DNA to to back to the viral RNA and produce viral DNA. Yeah, that.
So this direction.
Yes,
This direction.
This direction, then back to that.
Then back, yeah. Okay, that's good. And the last one, replication to divide the cell. What is that? To divide the cell, what is that? That's DNA dependent DNA polymerase. Okay, so now we have to go inside. This transcription is very important because that is what gives me what kind of protein that I have. So let's spend more time on transcription because we asked a lot of questions about those on the exam. Yes. I'm sorry? Yeah, this past exam we asked about the transcription. We asked about transcription, And we have to have so now we know what enzymes are implicated. Now we know what polymerases. So we end with ACE. By the way, most of the enzymes end with ACE, like catalase. Catalase, what is catalase? This one we cats. No, catalase.
Enzyme.
Yeah, it's to reduce oxidative stress. So when you have like in your body hydrogen peroxide, that's like really toxic. It will take hydrogen peroxide, convert it to water and oxygen. So that's a catalase. But end with ACE, end with ACE. But not every enzyme end with ACE, like hemoglobin. Is hemoglobin is hemoglobin
It does not end with.
is hemoglobin an enzyme? Yes, it's an enzyme. So before we talk about hemoglobin, let's see how we get hemoglobin. So we have DNA. Where is the DNA? It's in the nucleus. DNA is inside the nucleus. Then we transcribe the DNA, transcription using DNA dependent RNA polymerase. DNA dependent RNA polymerase. We use a DNA dependent RNA polymerase. And that happens in the nucleus to give me mRNA where? Nope. In the nucleus. This is still inside. In the nucleus. The DNA is nucleotide. The DNA is a nucleotide polymer. A nice, better way of saying that is it's a nucleic acid. And the mRNA in the nucleus is a nucleotide polymer. Nucleotide polymer. A nice way of saying that is that it's a nucleic acid. That's a nucleic acid. That's a nucleic acid. That's a nucleic acid. Where is that happening? Inside the nucleus. Then we go and we translate. We do translation. We do translation. Where does translation happen? In the cytoplasm. And in the cytoplasm, then you get your proteins. And proteins are two types. You have structural proteins. Excellent. And functional proteins. Structural proteins such as collagen. Where do we find collagen? In the ligaments of the bone and the skin. Where do we find keratin? That's the second one, keratin in the hair and the nails. Functional protein, that's like myoglobin, and hemoglobin. Myoglobin and hemoglobin. Okay, so if I want protein, there's no way I can get protein except through this way. You're never getting protein except even the virus, you know that. That's why he has reverse transcriptase. That's the central dogma of biology. You cannot change that. This is the Father, the Ghost, and these are the three. You can never change them. Do you understand me?
Sure.
That's the law. So, always when you think of a cell want to regulate some hormone synthesis, The cell want to make some protein channel that allows the entry of some electrolyte. The cell want to make some glucose transporters. Always will go to the DNA, transcribe it using DNA dependent RNA polymerase. Then the ribosomes will translate it. You know that protein channel? That's a protein. That's a protein. The protein channel to allow the entry of that's a protein. So we make that in the ribosome. The glucose transporter, that's a protein. Everything, hemoglobin, protein, all of the protein come this way. So there is a region for hemoglobin, region for protein channel, region for ion channel, region for cytoskeletal proteins, region for cilia, microtubules, region for microfilament. If you are a protein, you gotta go come this way. You gotta be born grandfather, father, that's him, that's the son. Is that point clear?
Yeah.
So now let's look at what happens in the DNA. So now let's say I want to make hemoglobin. Now let's blast the nucleus, make it big. And inside my nucleus, I have my daughter strands separated like that. I have 46 of them joined like an X. Like an X, I have 23 pair. I have either 23 pair of X or 46 that are similar. Now, all of this is inactive, off. The only on is what I need. So now I go for the hemoglobin region. This is my hemoglobin region. So that's my DNA, obviously. That's my DNA. And I have a region. My RNA polymerase will come, you know, the DNA is double helix. This double helix. So it will come with an enzyme known as helicase. Helicase. What helicase will do is it will unzip the zipper. It will unzip. It will unzip this zipper and separate it and open it up, exposing one strand. Okay? And exposing one strand. So we expose the strand and we're going to have our base pairs. Okay? We're going to have our base pairs. A, T, that's DNA. It has thymine. A, T, G, C, A. And you know the law. If you are an RNA polymerase, you're going to make proteins. If you are an RNA polymerase, you're going to make nucleic acids. You know that the DNA has guanine, cytosine, thymine, and adenosine. But the RNA, it has cytosine, guanine,
That's a U.
but for the, no, that's an A is fine, thymine, adenine, but
Oh, that one.
A, instead of a T, you will have a U. It will do just that. So it will make a transcript. For the A, U. For the T, A. For the G, C. For the C, G. For the A, U. And for the A, U. And it will, that is now my mRNA. That will give me my hemoglobin. So now this is all inside the nucleus. Remember, transcription, I didn't lie to you. Transcription happened where? In the nucleus. So transcription happened in the nucleus, mRNA in the nucleus. Now the mRNA that came out, let's say that this is the mRNA to give me hemoglobin. This mRNA is bad, it's premature, it's not ready yet. Premature mRNA. This is nascent mRNA. This is not ready. Premature, nascent mRNA. This premature, this nascent mRNA has to undergo, this is transcription, right? It has to undergo post-transcriptional modifications. What are the modifications? That's our mRNA. That's our nascent, immature, premature mRNA. And what I'm going to do is, like Agatha said, we're going to cut out some part that we don't need. So the premature mRNA has exons, 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 and introns, introns, exon, intron, intron, exon, intron, I'll come to this, exon, exon. So let's say that's exon A, exon B, exon C, exon D, and exon E. In order, that is my premature mRNA. That is my premature mRNA. That's my nascent mRNA that I need to do post transcriptional modification. So what I'm going to do now is I'm going to cut out what I need and leave behind what I don't need. Cut out what I need, leave behind what I don't need. So I need green. Green is what I need. I need green. I need the exon. I need the exon. I need the exon. A, B, C, D, and E. I don't need the introns. So the introns, I'm gonna cut them out. I'm gonna cut the introns out. I'm gonna cut the introns out. I don't want introns. So this is my premature, my mature mRNA. My mature mRNA is now gonna look like this. Yeah, exon A, gonna look like exon A connected to exon B, connected to exon C, connected to exon D, connected to exon E. Now that is my mature mRNA. When the ribosome read that, it's gonna give you the best hemoglobin, or it's gonna give you the best myoglobin. Yeah.
All right, so I understand everything you do. What I'm asking is, how do you determine that? Like, will it ever be like the intron will be the mature instead of the exon?
No, it's always the convention is the intron. Okay, so to make this more clear, this is still the exon. Where's the exon now? The mature exon now. This post-transcriptional modification happened where? Always in nucleus. This is everything nucleus transcription related. Everything transcription related is always inside the nucleus. That's where the students get confused because they say exon gets excised out. Exit is not, no. It is the introns who get left out. And they are called intron with an N because they stay behind in the nucleus. They're not going exit to the cruise, to the cytoplasm for the ribosome because they're not mature. It is the exon who will exit to the ribosome. It is the exons. The exon, this one, these exons, they call them exons because they exit from the nucleus to the cytoplasm. They go from the nucleus to the cytoplasm and they exit from the nucleus to the cytoplasm and they get translated. The introns don't get translated. They stay behind in the nucleus. Nucleus intron. So the intron stay behind in the nucleus. And the exon X exit to the ribosome. And then it's gonna get translated to give me my hemoglobin and to give me my myoglobin.
Join or they get these.
Yeah, so we're gonna get to that. The introns, now all of these introns piling up. Now all of them, if you leave them behind like that, they're gonna make stress for the cell. They're gonna be dumped, a lot of garbage, intron. So there's something known as alternative splicing. So in alternative splicing, we recycle, we reassemble them. We reassemble the introns, that garbage that we left behind. It stay behind the nucleus. It don't exit. It's not VIP. It's not going for ribosome to be translated. Don't worry. We're gonna reassemble them we're gonna recycle them, and we're gonna make them axon, and then translate them into some protein that's gonna somehow help us with something.
So basically, the introns, you're going to recycle them and make them,
Yeah, later on for some protein that is like gonna help with some secondary function, yeah. Yeah, yeah,
So this recycles the unused.
yeah,
Exon.
introns is gonna use the intron. So remember, introns with an N, they get cut out, they stay behind in the nucleus. It is the exons who exit and they get translated. Now I have this is my mRNA that's mature that I translated. To get my hemoglobin. So now we are up to translation. Let's translate hemoglobin so we can see the graph, how it works. Let's move on. Let's move on so we can see how the graph works. So now we transcribe hemoglobin. We did post-transcriptional modification. We cut out the introns. We left them behind. We did alternative splicing. We recycled them. Then the exons exit the nucleus. Now we're in the cytoplasm. So let's see now what happens in the cytoplasm. When we are in the cytoplasm, now, there are ribosomes. We said that, and this was a question on the exam. What happened was, if I have my final, but I do not read my final mRNA as exon A, exon B. It's in codons of 3 codons. The first one is always a start codon, known as AUG. AUG. That's the start. That's the start codon. AUG. Then you're going to have some other form like CCG, UCG, because remember it's gonna have U because it's an RNA. That's an RNA polymer. What's the mRNA? Is it a monomer or a polymer? Yeah, the mRNA. Is it a polymer or a monomer? Who made it? Who made the mRNA? Polymer, so it's a polymer. mRNA, yeah, compared to the DNA, it's small, but it's a polymer. Compared to Shaq, I'm small, but I'm still big. So this guy, mRNA, compared to the DNA, is small, but he's a polymer. He's definitely a big polymer. So this always gonna have AUG in the beginning. That's the start codon. You need to know this one, the start codon. And the start codon is a 2-in-1 because it's a start codon and it codes for an amino acid.
Methionine.
Who is that amino acid? Methionine. I promise you this one is going to be on the exam. What is the start codon? AUG. It also happens to code at the same time. Being a start codon, it also codes for what amino acid? Methionine. Then we're gonna have, these are codons. UGA.
Because can I just?
Yeah.
That one, 2 seconds.
Yes.
Yeah, no, I can do it.
Methionine. Methionine. And in the end, I'm going to have special codon called the stop codon or termination codon. Who are my 3 stop codons? You go away, you are away, and you are gone. Those 3. You are gay is not there. Okay, you are gay is there. Okay. All right. You are gone or you are gay. Okay. Those 3. You know, gay is happy. Gay is very happy. You know, back in the day when I was in elementary school, in 1964, they told us that there was a song He said, in a bright day, I was happy and gay, but now I'm not gay. I'm just happy and I am happy and because now gay means does not mean happy. So back in the day, bright day and happy and gay. But anyways, happy and gay, you are gone. Anyways, that's stop codon. That's stop codon. Does it code for any amino acid? I promise you that's on the exam. If I'm gonna ask you, the start codon. At the same time, it starts translation and it codes for another amino acid. What is it? Methionine. The stop codon, you are away, you are gone, and you go away. Those three, they also code for an amino acid. False. So they don't code for, they just terminate. So now these are codon. So that's my mRNA. That's my mRNA.
So you're gonna change a few questions, that's what you're saying.
These were on the exam, right?
Yeah, I just wanna make sure that.
Yeah. Yeah, just replace it a little bit. Just replace it. Okay. So the exam I'm gonna give you is gonna be strictly based. So they're gonna be 50 questions based on the 100 questions that were on the exam. Just make sure you solve that study guide and upload it by Tuesday, and then just work on those that exam, and you will see that the 50 questions we're gonna have, each one is gonna work 2 points, and that's going to be like strictly based off that exam. So just changing the questions a little bit, but it's strictly based on it.
Making them smaller and the T.
Okay.
More.
Okay, yes.
Two things. So the first thing I wanted to know, are we uploading both study guides, study guide 1 and 2?
No, you just, so for one, so I'm gonna delete that old portal,
Gotcha.
or maybe it's the same portal, or it's not gonna allow you to re-upload, right? So I'm gonna delete that whole thing. Okay, so I'm gonna edit the date. Can you delete the one you post already?
You can resubmit it, but you're not gonna.
It lets you resubmit. Okay, so we'll do that. I'll extend the date and you replace it there, just with the numbers, no question. Just put your answers in there. And just describe the answers.
And both study guides, both.
And one. So-
Oh, okay, say that, I got you.
Yeah, assemble them together into one and then submit it. But the exam is not gonna be 100, it's gonna be 50. But based off of that exam. Yeah, I'm sorry? Oh, the phone guy. Okay, all right, we're gonna switch to the. Yeah. Oh, thank you very much. Yeah, I try. Thank you, I'll try. I'm gonna do that some more. So I'm gonna do my best. You know, we have to be problem solver. It's nothing personal. Nothing personal. We always have to work together so that we all gonna climb the mountain. Both of us in the same trouble. Both of us, we have to climb. Okay, other courses. No, we're not done. You have half an hour. Please, let's finish that. Let's finish translation. Okay. So I'll give you a break in the end.
We don't need no break, we don't need no break.
Okay, so let me just finish the translation and then you can go home. Please pay attention. So now that the start codon, who binds is the small ribosome subunit. The small subunit of the ribosome is gonna bind to the bottom, and then the large subunit is gonna come in, and it's going to have 3 different sites: EPA, E for exit, P is for polypeptide, and A is for entry. And then you will have, that's a rRNA, that's a ribosomal RNA. Is that a polymer?
Yes.
Yes, it's RNA is a polymer. rRNA, polymer. mRNA, polymer. Then the tRNA is like a clover leaf. A 3-leaf clover, or a clover leaf, or whatever that is like that. It's going to come on its behind, you will have the amino acid, on the front, it will have the anticodon. So for example, methionine will be in the back, and in the front will be the start codon. So A is gonna bind with U is gonna bind with A, G is gonna bind with C. So that's gonna be like this. UAC, they're gonna bind here. Methionine is gonna come in the entry site, then it's gonna move to the polypeptide site, and the next tRNA is going to the transfer RNA. This tRNA, three-leaf clover, is gonna come here, is gonna bind, and then a shift will happen. Methionine is gonna shift to here. So now we're gonna have 2 amino acids. Then they're gonna move to the P site. Then the third tRNA is gonna come in, and they're gonna shift shift is always from P to A. So now you're gonna have 3 polypeptides. Then they're gonna shift. When you shift, they go to the exit, they leave. Then this guy comes here with its 3 amino acid. Then the fourth tRNA comes in, and they shift from P to A. You're gonna have 4 and so on until we hit the stop codon. Then you're gonna have your long string of beads. That's the primary sequence. And then the secondary sequence, that's gonna be based on hydrogen bonding and hydrophobic effect. You're going to have alpha helix and going to have beta pleated sheets, alpha helix, and beta sheet. Then you're gonna have tertiary structure, and that's gonna give you your myoglobin. And you got that because of more extensive hydrogen bonding and hydrophobic effects. And lastly, you're gonna get your quaternary structure, hemoglobin, due to more hydrogen bonding, more hydrophobic effect, and more ionic bonding, and that's your hemoglobin. So what did Christian Anfinson do in the experiment? He realized that when you heat up the enzyme, or when you play with the salt, and when you play with the pH, it's gonna unfold. Why? Because you destroy the weak bond. Ionic bond destroyed, hydrogen bond destroyed, hydrophobic effect destroyed, van der Waals destroyed, but the covalent bond is not destroyed. That's your primary sequence that came out of the ribosome. So they fold backward into the primary. Then when you restore the optimal condition, they refold back up to the quaternary structure. So he discovered that it is the primary sequence. That's the manuscript. That's the one that dictates how the folding will happen. And remember, myoglobin is tertiary, it's good enough. It doesn't need to be quaternary. It is the hemoglobin who is quaternary. Yes?
So, real quick, I get all of that, except can you repeat back from the ribosome, how we go
Okay.
into the first primary?
This, we go to the what?
The ribosomes, when you know from the A to the P. to the
Okay,
X.
this one you need to see a video. I'll show the videos. This one because you cannot see it except it's happening. So I'm gonna post it. But remember, that's my Christian Anfinson question. Then the last thing before you go is the graphs, the board graph that showed us. What did it show us? So ferric Fe+2. That is what binds my oxygen. Ferrous is Fe+3. So let's look at my graph. My graph is like that. What is it showing me? My graph is showing me oxygen. This is really important. I wish the camera was working. Oxygen saturation. This is oxygen saturation. Or oxygen affinity. Affinity. Hugging oxygen. And this is partial pressure in torr, partial pressure of oxygen. Excellent, Gaga. This is partial pressure of oxygen. So we have 3 separate molecules: hemoglobin, adult hemoglobin, fetal hemoglobin, and lastly, myoglobin. Okay. Let's begin with myoglobin. Myoglobin storage. If myoglobin and hemoglobin both holding oxygen, they come by a tissue screaming for oxygen, myoglobin will never give oxygen. It is hemoglobin who will give the oxygen. The myoglobin will let it die. So that's storage. So myoglobin is hyperbole. Look like that. Myoglobin is for storage It's a storage enzyme. And it has high affinity for oxygen, diatomic. Oxygen is always O2.
What's the word that you use, a specific word?
Diatomic.
No, high something. High, say it again?
Hyperbolic. Yeah, this form is like a hyperbola. This shape right here is hyperbolic. Now the other two are your adult hemoglobin. That's orange. It looks like that. That's sigmoid. Sigmoid. And that's reflect that it is excellent for transporting oxygen. It's excellent for transporting oxygen. It's excellent for transporting oxygen. And then there is fetal hemoglobin in the middle. That was one of the questions I asked you to put the order. So fetal hemoglobin is here. That's fetal. It's also sigmoid because it's hemoglobin. That's fetal hemoglobin. So going out this way, that's the order of increasing affinity. Nobody holds on to oxygen more than myoglobin. So the one who holds a lot of oxygen, that is myoglobin. Fetal hemoglobin also holds oxygen more than adult hemoglobin. Adult hemoglobin is the one that has the least affinity. So when you put them in order, the most affinity, myoglobin, because that's a story, that has nothing to do with transport. Then the baby will hold on to more oxygen more than adult, and then adult is the least. So now what did Bohr discover? Bohr discovered when you have nasty tissue. So normally that's normal condition. According to this partial pressure, remember I told you this, think of this partial pressure as percent of oxygen. That's 10, that's 20, that's 30, that's 40, that's 50. So at here, this graph shows you that if the partial pressure is 10, hemoglobin will only be saturated with 90% oxygen. Hemoglobin will be saturated. So when there is only this is the partial pressure. So when it increases, that means I don't need oxygen. So hemoglobin will have high affinity. So hemoglobin will have high affinity in tissue that doesn't need oxygen. So if you're healthy, your skeletal muscles are relaxed, you're watching the Super Bowl, hemoglobin is not going to give a lot of oxygen to your legs. It's going to give a lot of oxygen to your stomach because you're eating and it's absorbing. It's need oxygen. So over there in your stomach, the partial pressure for oxygen is low. There's not a lot of oxygen, so we will give it there. But the partial pressure for oxygen in your leg is high. The partial pressure for oxygen in your lungs is always high. You take a deep breath. Oxygen comes in the lung. Oxygen comes inside the lung, then hemoglobin will take it. It will not give it to the lung because it has high. It will give it to area where there is low saturation. So the affinity for oxygen in hemoglobin in low partial pressure will be low. Affinity means holding on to oxygen, storing oxygen. So hemoglobin, when it sees low oxygen, will give it off. So it has low affinity. It has low affinity for oxygen in low saturation. But when the saturation is high, it will have high affinity. That's why that's the saturation. At very high, so in tissue that has 100% oxygen, like the lung, that's the lung. When the tissue has 100% oxygen saturation, the hemoglobin will be saturated with a lot, with 100%. It will be saturated because it doesn't need to give it off. But when the saturation is so low, like 20%, you're running a marathon, the affinity will be so low because hemoglobin gave off all of the oxygen.
That would be myoglobin, right? The orange, correct?
Yeah, I'm sorry, I meant here.
Oh,
I mean here. But the point, you get the point. Affinity is high when the oxygen saturation is high, like the lung. Hemoglobin will have high affinity for oxygen in the lung because the lung doesn't need oxygen. but it will have low affinity in the legs when you're running the marathon because the partial pressure for oxygen is so low.
So I just want to make sure hemoglobin, low affinity for oxygen and saturation.
Low affinity for oxygen in areas that have high saturation, that have low saturation of oxygen.
Yeah. Okay.
Yeah. So an area that has low saturation of oxygen,
Has low,
saturation of oxygen, because you consume it. Why do you have low saturation of oxygen? Because you're consuming it. You're running the marathon. Your legs are consuming oxygen.
yeah, that's all right.
So the oxygen saturation in your legs will be so low because you're running the marathon. The oxygen saturation in the legs is going to be so low. So over there, the affinity
Low,
for hemoglobin is going to be low. The saturation for hemoglobin is going to be
low.
low. It's going to give it all. But in your lung, you're breathing, there's a lot of oxygen, the saturation is going to be high. Affinity is going to be high. That's not what Bohr said yet. Bohr said, in addition to all of that, the nastier the situation gets, the more acid there, the more 2,3 BPG, the more acid that's there, the more 2,3 BPG that's there, the more carbon dioxide that's there. Hemoglobin will let go even more oxygen than before. So when you are in these normal situations, when the saturation, for example, is 20 at when the oxygen in tissue is 20, hemoglobin will only be saturated with 20 because it will give off 80. But if the situation gets worse, the graph is going to shift right. So that means it's going to be at situations where the partial pressure is less and there is more acidic and more carbon dioxide, hemoglobin is going to be even more unsaturated, even more less affinity. It's going to let go of oxygen more. So that makes a lot of sense when we have the graph in front of us. So now I think you guys want to go. I don't want to hold you more than that. You guys want to go, but make sure. Okay, so now with this background information, attempt the study guide. Don't worry, this is not classified White House information. This is all in the videos, YouTube, is in the book, is in the PowerPoint. But I'm gonna make that review session again and post it for you over the weekend. Just make sure that you don't just sit down, wait. Keep looking, keep Googling, keep searching. You know when you swim, when you're in the water jumping, you're gonna start swimming. But don't wait there. I'm working with you, but you have to also keep searching, keep searching. When we meet next time, we exchange. So next time when we meet, we're gonna have the exam and we're gonna start the metabolism. But I'm gonna keep going back to that. I'm gonna keep going back to that. Yeah, let me take you 10 minutes, okay? So don't worry, we're gonna be working on. Yes.
So I just want to make sure